Hi everybody, this is section 20.2 and we're going to be talking about the polymerase chain reaction, the 3 steps of the PCR cycle, and then we'll also be talking about reverse transcriptase PCR and how that's carried out, and real-time PCR and why it is used. And reverse transcriptase PCR along with real-time PCR, we're actually seeing that with coronavirus as a way to test for it, and so hopefully this will give you kind of a preview into that. And for those of you who were in lab, hopefully this is a good refresher of what we actually did in the lab with our salmon samples. So the polymerase chain reaction is a way to make many copies of a particular gene DNA sequence. And this was developed by Carey Mullins in 1985, so it's actually fairly recent. I guess that maybe dates me if I say that's fairly recent. But this actually revolutionized the way that we look at DNA and the ability we have to do studies of it. And that's because, unlike gene cloning, PCR can copy the DNA without any host cells or vectors. It's just copying those actually double-stranded DNA into many, many copies, and it can be done in a very short amount of time with a thermocycler. So the way that we do this is if you have a particular chromosomal DNA, so this could be a whole chromosome, and we have one particular little section that is our gene of interest, we can actually take a primer that can bind at either end of that gene of interest, and we can go through and make many copies of this particular gene of interest by going through a bunch of polymerase chain reaction cycles. And this can happen all in a thermocycler with the proper things that are added, proper ingredients. So the starting materials, what you need for PCR, and we put this in very small microcentrifuge tubes. You start out with your template DNA. So whatever that DNA is, whether it's maybe a particular, maybe it's the cDNA from coronavirus that you've made through reverse transcriptase, or in the case of the salmon, perhaps this is our salmon DNA that we isolated, right, that we extracted and purified. Then we have to have primers, and these are complementary primers to the sequences at the end of that fragment. So keep in mind, when we have our entire template DNA, we're looking at just a region within that, and we need to have these primers that are complementary to the sequences that basically bookend that one section of the DNA that we want amplified. So in the case of our salmon, we had the entire genome of salmon, but we only were amplifying a segment or fragment of DNA that was 650 base pairs long. And so keeping that in mind makes more sense as we think about it, that it's just one section of the DNA. These primers are synthetic, so they're made in laboratories, and they're usually about 15 to 20 nucleotides long. And so you need to know the sequence of the DNA to be bound by the primers. Now, in the case of coronavirus, obviously we didn't know the whole sequence, but we did know enough about it that we could come up with synthetic primers that would match up because it had similarities to known viruses. We also need a lot of deoxynucleoside triphosphates, so these are our DN-to-Ps, these are our A's, T's, C's, and G's that need to be added in to make the new strands of DNA. So think about what you have to make DNA. We need the nucleotides, right? And then you need to have a DNA polymerase that is stable at high temperatures. And this is necessary because in PCR, we use heat as a way to inactivate other DNA polymerases, but also to separate the strands of DNA to allow us to have templates available to make new strands. And so in this case, we generally use what is called Taq polymerase. This comes from a bacterial cell that is, I believe it was it was found in Yellowstone Park in some of the hot pots over there. It was a bacteria that could actually grow at very high temperatures, which means its DNA polymerase can work at very high temperatures. And so what we end up doing is we have our DNA strand of interest. We need to do denaturation, basically taking the 2 strands and separating them so that we have a template to begin to synthesize a new one. We need to do an annealing period where we bring in our primers that can bind and bookend that section that we're most interested in. And then what we do is we allow that polymerase to synthesize or extend those primers to copy that target sequence And this all takes place in a thermocycler. Before thermocyclers, this was a very tedious thing that had to be done in the lab, but now our thermocyclers can actually regulate these temperatures in cycles and allow this reaction to occur over and over again in a matter of hours. So this is showing you kind of a figure of how this would happen. So you have your template DNA, and we have one particular site, perhaps it's from here to here, that we're super interested in. We have primers that we know the bookends of the gene of interest, and so when we do denaturation, what you see is that our two strands are separated and we have two templates ready to have a complementary strand made. These primers that we've added in will bind to the part that they are complementary to, and then we can go through primer extension. And this is where we have the Taq polymerase that will build those complementary strands. So when we look at this, we want to keep in mind the temperature. Denaturation takes a high temperature to separate these two strands. Then when we can lower the temperature and allow the binding of our primers. And then lastly, we raise that temperature again a little bit in order to have that DNA synthesis occur with the Taq polymerase. And that is how we end up getting these two DNA strands now, right? We started with one and now we've got two that have our gene of interest. Well, what's nice about this is that a typical PCR, polymerase chain reaction in a thermocycler, involves about 20 to 30 cycles of replication, and this only takes 2 to 3 hours really to complete. After 20 cycles from our original DNA sample, we will increase that of 2 to the 20-fold, so about 1,000,000-fold. And if you run 30 cycles, it will be 2 to the 30-fold, which means that you will have around 1,000,000,000-fold of your interest, your gene of interest. This is showing again what this looks like. Up at the top is cycle 1, so you have your first one that then denatured and we got 2 strands from. We go through the exact same thing, the denaturation, the adding of the primers, and then the expansion of that. So you start out with our 1 strand, we then go to 2, the next cycle goes to 4, the cycle after that goes to 8, and the cycle after that goes to 16. So you can see how quickly this is, because with each cycle, what we end up doing is we end up getting a doubling of whatever we had from the last cycle. So this is a very quick way to have a large amount of the particular gene we're interested in. The really nice thing about this is as we start going through it, we are going to have more and more that are just the primer flanked gene of interest, because what begins to happen is that we have them that have shortened on either end, and what ends up happening is that out of your million or billion copies you make, there will be very few of these longer ones, and the majority of them will be exactly that gene of interest flanked by your primer. So we can also do what is called reverse transcriptase PCR, and this is a way to look at quantifying the amount of RNA in living cells or in viruses. And so the method is reverse transcriptase PCR. And what happens here is a little bit different in that we isolate the RNA. So again, think about the coronavirus. We take that virus and we isolate it, and then we mix it with deoxyribonucleotides and reverse transcriptase it. And so in this case, what you're going to end up with is first a single-stranded cDNA, right, is produced that's complementary to that RNA, and then that first single-stranded cDNA, right, complementary DNA, will work as a template now to go into conventional DNA PCR. And so this RT, this reverse transcriptase PCR, is extraordinarily sensitive. It can detect very small amounts of RNA in a single cell, and this is in some cases used to actually look for the coronavirus. And so this is giving you a picture of what this might look like. Here's the RNA, so perhaps this is the genome out of coronavirus. We add reverse transcriptase and a primer that can bind it near the 3' end, and then we add those deoxyribonucleotides. And what you get is, rather than a complementary RNA, you get the complementary DNA. This can go on to that conventional PCR and give you a large amount of the cDNAs, of those complementary DNAs to that RNA of interest. We can also do something called real-time PCR, and this is looking at the amounts of product that can be measured in real-time PCR process and not just at the end. And so this can give us a way to compare amounts of DNA template, and we can look at the amount of product as it correlates to the amount of target that we originally had in the sample. In this case, you use that thermocycler that can measure fluorescence from the probes. And so if we see an increase in of the probes, the fluorescence, we know we have more of these being made. So for checking in on this, I'd like you to think about what the goal of PCR is. Also, why does the DNA polymerase need to be effective at high temperatures? And then thinking especially about coronavirus, what is the benefit of this RT-PCR? How does this aid us? So hopefully this helps on the PCR and a good review for those of you in lab. Please let me know if you have any questions on this. Thanks.